In the last lecture, we studied about the cell. But how does the details about the cell came into our knowledge? That's where microscopy comes in. The term micro means not visible to the naked eye, and the scopy means to study by instrument. So, the microscopy is a technique used to visualize objects that are not visible with naked eye. The instrument used is called microscope. Now, there are two terms related to microscopy. These are magnification and resolution. The term magnification means to increase the size of object, that is, a small object is made seen as a larger one. However, the term resolution means to see two closely placed objects as two separate objects. There are two types of microscope, the light microscope and the electron microscope. A light microscope uses light to visualize the objects, whereas an electron microscope uses an electron beam. Electron microscope is of two types, the transmission electron microscope and the scanning electron microscope. In a detailed view, a light microscope is what we usually see in our labs. It has an eyepiece or the ocular lens. This is where we visualize the objects. This piece comes in contact with our eyes. That's why it's called the eyepiece. Then we have an objective lens that actually see the specimen. Then we have an arm to hold the object. Then we have a stage on which the slide is placed. Below the stage we have a condenser, and in the bottommost area we have the light source. Now, to understand the working, light source releases light beams that are received by the condenser. Condenser condenses them into a narrow beam. Condenser also has a diaphragm that controls the amount of light coming to the slide. The light strikes the slide containing the specimen. Then these rays are transmitted to the objective lens. This is where the first image is formed. It is the real but inverted image of the object. This image has been magnified. Next, the objective lens transmits these rays to the eyepiece. This is where the second image is formed. It is a virtual upside image with a very large magnification. It's just like the image is formed in our brains. Like the first image is formed on the retina that is inverted, and the second image is formed in the brain that is upside down. Now, talking about the electron microscope has an electron gun, an anode, condenser, objective lens, projector lens, and the screen. The screen is where the slide is placed. The slide transmit the electron gun acts as an electron source. It transmits electrons that are accelerated by the positively charged anode. The condenser condenses the beam of electrons, that is, it narrows down the path, transmits them to the objective lens. The objective lens transfers them to the slide. As soon as these electrons strike the object, the different rays are emitted in different directions. These rays are converged by the eyepiece and the objective's view. The differences between light and electron microscopes are that light microscope has a smaller magnification. It can magnify objects up to 1500 times. However, an electron microscope can magnify objects up to 1 to 50 million times. Resolution of light microscope is very small in as compared to the electron microscope. The specimens that are observed in the light microscope can be live or dead, but the electron microscope only observes dead and dried objects. Light microscope produces colored images, whereas an electron microscope produces black and white images. Science fact: an eyepiece of a light microscope has a magnification of 10x, that is, it can magnify objects 10 times. However, we have three different types of objective lens. They have magnification of 10x, 40x, and 100x. The overall magnification is the multiple of both the magnification. For example, if we observe an object in this 40x and 10x, then the total magnification will be 40x multiplied by 10x, that is 400x. That's how we find the magnification in the light microscope. To summarize, smaller objects are seen by a technique called microscopy. The instrument used is called microscope. There are two types of microscope, the light microscope and the electron microscope. Light microscope uses light source to visualize the object, whereas an electron microscope uses an electron source. Both the lenses have a condenser that narrow down the path of light or electron. The object is visualized with the help of eyepiece. The magnification and resolution of light microscope is low as compared to the electron microscope. Live and dead specimen can be observed in the light microscope. However, electron microscope can only see dead and dried specimens. Light microscope produces colored images, whereas electron microscope produces black and white images.